to be here.
Sorry.
It's a pleasure to be here. And first of all, I think we should be on a first name basis. So um, you can call me Roger. I'm going to spend some time comparing the purposes, content, and sort of necessary components of two key documents to getting a clinical trial funded and actually being able to run it. And most of my point is going to be that these documents have very, very different purposes. So any attempt to use text or materials commonly between the two documents is likely to get you into trouble. And that's my theme. So these are my disclosures. Importantly, I am a part-time employee of Berry Consultants, so that's a statistical consulting firm. I checked my email this morning. To the best of my knowledge, I'm still a special government employee. I suspect that they just haven't gotten to me yet. And also, last I knew, we have support from NIH. So there are lots and lots of different documents that may be required to get support and to run a clinical trial. And these documents have different purposes, and not all may be required depending on the details of your trial. First, if you're trying to get funding from a granting agency, then obviously there's a grant application, and that's a document that's submitted to and reviewed by funding agencies. And the goal of that document is to secure grant funding. There's a very important distinction between grant funding and having a contract. Grant funding is a mechanism of transferring resources that gives much greater autonomy to the recipient of those funds to do what they see as best with them. So in contrast, you have something called a proposal, and by the way, grant application and proposal are not synonymous, which is a document submitted to and reviewed by a source of financial support or its proxies, but the goal is to secure a contract for proposed work. A contract is a much more binding or restrictive relationship with your funder. You may absolutely have sorry about that. You may absolutely have a contract from a federal funder. They do have contract mechanisms. There's also other funding mechanisms, but just be aware that with a grant, you're given autonomy to run your program within reason. With a contract or some other cooperative agreements, you're doing it with them and you're partners in the activity. A clinical trial protocol is a document that details the design, the conduct, and the planned analysis of a clinical trial. It is intended to help people run the trial consistently and as you intended. Usually that clinical trial protocol does not include all of the nitty-gritty details of the statistical analysis plan, the design, what the interim analyses are going to look like, the final analysis, handling of missing data, all sorts of details. Those are usually captured in a statistical analysis plan, but not always. Sometimes people try to fit all that stuff in their protocol. But the statistical analysis plan is a comprehensive description of all of the statistical aspects of the trial. Other documents that may or may not be required for running a trial are a manual of procedures, sometimes called a manual of operations. That is a document that gives the real nitty-gritty details of how you do things. You know, if there's a consent procedure that says, what do you do if the patient is unable to consent and the next of kin are not available? What are your next steps? How do you document things when you draw the blood? How do you spin it down? Which color tube do things go to? Who do you mail or ship the specimen to? Details that would be very distracting were they in the clinical trial protocol. And moreover, those details may change very frequently. The address you ship it to or the name of a contact may change. You want to be able to frequently update that in the manual of procedures without having to modify your protocol, which could trigger a re-review, for example, by your IRB. So the manual of procedures is this flexible living document where you keep track of how you're doing things. It is also intended to help, for example, in a multi-center trial, everybody do things the same way. The DSMB charter is exactly what it sounds like. It's a document that talks about the role and function of a data and safety monitoring board or data monitoring committee if your trial requires that. What we're going to do is we're going to focus on these two documents, because what we've seen is that as people prepare materials for this course, sometimes we're asking for things that might go in a grant application, sometimes in a clinical trial protocol. And if there's any fuzziness in that distinction, it can lead to wasted effort or people sometimes not getting off on exactly the right first step. So as a general rule for a grant-funded clinical trial, both a grant application and a clinical trial protocol are required, and neither are sufficient. And what I mean by neither sufficient, other than the obvious interpretation, is that sometimes people, when you ask for a protocol, send you their grant application because they've actually never written a protocol. And I will tell you, it is not a substitute. And the reason it's not a substitute is because the purpose, the audience, the content, everything is different about them. So for the grant application, the purpose is to secure funding. It's to convince somebody with money that investing their money in your research program is a good investment for science or to further their goals, whatever they are. The target audience are generally expert review panels and funding decision makers like study sections or NIH councils or their equivalent. These are technical, informed people who are comparing, in general, multiple highly technical documents. The content is intended to support the decision to fund your research, so the background must be very sound. You clearly identify the gaps in understanding that your research is intended to fill and why those gaps are important, the rationale for the approach you're taking, the specific aims, how you're going to do it, but also a lot about why people should believe that you're going to be able to successfully do this, the team qualifications, and that it's a good investment. The budget is reasonable, the costs are justified, etc. Because these documents are intended to be part of a fair but competitive process to seek funding, there are rigid structures and page limits. I mean, you've all seen this. You're allowed exactly this many words or pages for different sections. In contrast, the clinical protocol is a document that becomes most important once you've received funding or you're planning to launch your trial. And its purpose is to precisely define how you'll actually do the trial. And its target audience is completely different, and in fact, in my view, much broader. So it's the study personnel, which may be research associates, it may be study coordinators, it may be faculty members, reviewers, including lay persons on the ethics or IRB review committees, or even regulators. It is narrowly focused on how you're going to do the trial, whereas the trial, when it's described in your grant application, is just a component of the scientific program for which you're hoping to receive funding. The protocol will give a brief background, but then talk about the overall design of the trial itself, the objectives, how you're going to enroll patients, consent them, collect data, monitor adverse events, and analyze the data, etc. And obviously, these lists are not meant to be exhaustive. But the details of how you're going to do it are going to be in the clinical protocol to a large extent. Because the complexity of a clinical trial varies widely, the lengths of these documents, clinical protocols, vary widely. But I would say typical is 30 to 150 pages. If I see a clinical protocol that's 12 pages, I think someone has tried to take their grant application and quickly edit it and try to call it a protocol, and it will likely have substantial holes. So here's a comment that I just found on the web that sort of further underscores this distinction. It says grant writing is about getting the money. Science is what you do when you get the money. They're two distinct activities. First, get the money. Second, decide on the best science to do with that money. Getting funded and science and funds are not the same. This is from a basic scientist, a cell biologist. Um, and this is obviously for a grant mechanism where once you get the money, then you decide what experiments you're going to run or exactly how you're going to do it. But the grant provided enough detail so the study section felt that this was a good investment, that you were going to be able to further the science that was of interest to them. So let's look a little bit at outlines and how they're both similar and different. Although I will tell you that the similarity is sort of almost like a false cognate, where it can lead you to think things are the same when they're in fact not. So what I did is I took just an online outline of the research plan part of an NIH clinical trial application. So this is the PHS 398 part. And this is selected parts. I've excluded the budget human subjects. And let's talk a little bit about what people are looking for in these different parts. So the introduction is where you place the research that you plan to conduct in the greater context of the field. You establish its importance. You identify the clear gaps in current knowledge that you intend to fill, and why filling those gaps furthers either the scientific or health mission of the research program. That should leave the reader with a clear understanding of the current state of the field and what we need to do to make the next substantial step forward. You then lay out a set of specific aims, and we're going to talk a little bit more about them in a second, but these are a set of interrelated things that you're going to achieve, all of each one of which will help in some way further us towards plugging the gaps in knowledge or filling the gaps in knowledge that have been established in the introduction. The research strategy is a description of how you're going to do it, but because of the page limits, it's by necessity inadequate in detail to allow someone to conduct the research that you intend or to run the clinical trial that you intend. So the research strategy has to be carefully structured to provide enough detail so the reader can impute that you know exactly what you intend to do and you will be able to do it without giving them all the detail necessary to do it. And that is absolutely an art. When I read a research strategy, when people add sentences about a pitfall and how they're going to overcome it, that shows me that they've been there and they know the solution, that's really helpful, because I gather from that that they have a realistic understanding of the challenges that they're likely to face. A progress report or publication list to help establish that you can be successful in this field, that you can be productive. This also is a proxy not only for your own abilities and work efforts, but also the fact that your environment actually allows you to be successful. You have enough protected time, etc. When there's multiple PIs or project directors, how is the leadership going to be structured among them? Do you have a management plan, contractual arrangements, letters of support? I want to spend a second on letters of support. You should be seeking letters of support many, many weeks or even months before the due date for your grant. It is not the last thing, because we could ask the other faculty in the course, but when I look at letters of support, if it looks like everybody got the same draft Word file, changed a few words and signed it, I consider those as either null or negative with respect to evaluation of the grant. The letters of support need to come from people who really have insight into what you're doing, and each letter needs to reflect their individual insight and support for the importance of or your ability to complete the work. So please don't clone letters of support, send them out a week before the deadline, and ask everybody to put it on their institutional letterhead, sign it, and send it back. Resource sharing plans, other plans appendix. So this is the general, some of the key areas. In contrast, a clinical trial protocol, as I said, is much more narrowly focused on how you're going to do just the trial. And the trial is part of your research program for your grant. It is not the only thing in it. So the title page, which also includes the name of the principal investigator, version control numbers, and those sorts of things. The background and rationale here, which is somewhat analogous to the introduction or background in the grant, is much narrower. So the background and rationale can be drugs in this category X have been of considerable interest in the treatment of Y, and we think drug Z is particularly promising. Drug Z has been used in up to 22, healthy volunteers without adverse effects, and therefore we plan a trial to see if it works. That's it. The study objectives, hypotheses, and endpoints are kind of analogous to the specific aims, but they are much more direct much more limited and directly related to the endpoints of the trial. The overall study design, this is a randomized double-blind placebo-controlled trial. This is a randomized open-label pragmatic trial, whatever trial design is. And then you get into how are you going to do it? How are you going to enroll participants, screen them, decide if they're eligible, informed consent processes. I think someone's the intervention and treatment. How are you going to administer the control, the intervention treatment? What outcome measures are you going to collect? Both primary, secondary outcomes, safety outcomes, timing, etc., definitions of adverse events, procedures for recording them, the reporting requirements for adverse events, then a separate section on the overall statistical design, which I will say is usually sort of medium detail in the clinical trial protocol, how you arrived at the sample size calculation, and then usually a separate section on ethical considerations. Informed consent does need to be mentioned in the enrollment part, but sometimes there's more, there's additional considerations regarding the informed consent process or the IRB review process that's there. For example, the use of a central IRB. But one of the things about that I was concerned about in showing two outlines is that you can get this idea that for most things on the left, there's sort of something on the right or vice versa. But even within things that look similar in focus, the level of detail is markedly different. So as I mentioned earlier, the scientific background in the grant needs to show an extensive understanding of the state of the literature and prior work in the area. In the protocol, it's just enough to basically put into context the trial. The specific aims and objectives need to be very detailed in a grant. They are usually simpler in a study protocol because an objective is actually simpler than a specific aim. The overall research strategy, because it's a main part of why someone should believe you might be successful and should fund you, is usually quite detailed in a grant. We usually spend just a paragraph saying what is the overall trial design in a protocol. A lot more detail about the clinical trial itself in a protocol, whereas in the grant, you're just trying to describe it in enough detail so people believe you know what you're talking about and can do it. The statistical analysis plan in the grant is freestanding, and usually what I would call a moderate amount of detail, enough so the statistical reviewer on the study section understands and agrees with your design, or at least can support it in discussion. In the clinical trial protocol, there's usually a medium amount of statistical analysis, but nowhere near enough for a statistician to actually do the coding to analyze your trial data. That level of detail is in the statistical analysis plan. If there are interim analyses or data monitoring, for example, checking data partway through to see if you can stop early for success or for futility, those are usually briefly described in a grant because the purpose is to tell the review team that you will not continue this trial and spending their money if it no longer makes sense to do so. In the protocol, it's similarly described, but the SAP, where the statistical decision rules are laid out, need to be precisely defined so there isn't second guessing whether any of those statistical triggers for early stopping have been met. Again, in the grant, you're going to point out that you'll collect adverse events and perhaps name specific adverse events that are of interest for the type of intervention that you're studying. In the protocol, those procedures need to be very detailed so there's no ambiguity regarding whether a particular type of event needed to be reported or was reported properly. And with the grant, if you have a statistical analysis plan, and I should say if you have a protocol, and there is a mechanism in the grant mechanism to allow you to include those as appendices, you should absolutely include them. You always want to show them that you've thought out the details, even though the details can't be put into the page-limited research plan. For the protocol, the SAP, the DSMB charter, and the manual of procedures are separate documents or they're appendices, but they all are part of the trial master file, so they're all considered part of the total corpus, if you will, of documents that govern the conduct of the trial. So at this point, what I would like to do is I would like to come back to this distinction that I think actually is the one that trips up our students, and I have to say our faculty as well, more than any other. And that's what is the difference between a specific aim in a grant and an objective statement in a clinical trial protocol. So if you go back to the concept that a grant is intended to get money, then you're asking someone to use their scarce resources to support your scientific program, and you want to show to them that you have a set of very well considered steps or components that build up this entire program that they are investing in. It is generally a broader set, and what I mean by broader is broader relative to the objectives that I'll show in a second. There need to be very precisely defined and achievable goals with clear criteria for success. A lot of things that you will read will say each specific aim needs to have a hypothesis. I think that's overly restrictive, but there needs to be clear criteria for success. Sometimes that is demonstrating a hypothesis to be true or demonstrating a hypothesis not to be true. But sometimes the clear criteria for success is, for example, the establishment of a research consortium with at least 20 members and this patient volume. Sometimes it is the successful, isolation purification of a compound, which may or may not be a hypothesis per se. There is a very important distinction between interconnectedness of the specific aims and being interdependent. Interconnected means they all are part of a broad vision regarding the goals of your research plan. Interdependent means if I fail at one, the whole house of cards falls down. The first is good, the latter is not good. So you need to be very clear whether or not any of your specific aims can only be pursued and completed successfully if a prior one is completed successfully. You don't want to be ambiguous about that, and you want to make sure that if there's an aim that does not have a very high chance of success, that you have a plan for what you're going to do to preserve the value over the overall research program in light of failing to achieve one of your specific aims. Sometimes specific aims are focused on demonstrating principles. I'm going to demonstrate that when I administer this drug, it actually gets to the target, or there's target engagement, as opposed to a patient-centered efficacy measure. And then lastly, the objectives when you're seeking support for a clinical trial, the objectives of that trial should align and may be essentially identical to one of the specific aims in the grant. So the specific aims include things that are not objectives of the trial, but the objectives of the trial should tie back to the specific aims. I'll come back to that right now. So in contrast, the objectives are much narrower and directly in focus. And in general, I like every objective in a clinical trial protocol to be tied to a specific analysis of a specific endpoint. So it's to demonstrate efficacy on this endpoint. It's to demonstrate decreased pain. It's to demonstrate a lack of thromboembolic complications. Whatever those objectives are, they're tied to an endpoint. You should never have process goals be an objective of a trial. An objective is not to, engage X numbers of sites or this number of investigators. It is the objective upon completion of the trial. Whereas in a specific aim, you may have specific aims that are process related if those are particularly important with respect to the overall program. So the last bullet here is just a mock-up of a clinical trial objective to demonstrate the efficacy of the treatment compared to placebo on an outcome after a specific period of time in patients with some disease. Okay, so that could be an objective. That could be the primary objective of a trial. That objective should be related very closely or mapped to at least one of the specific aims of the grant seeking support for that trial. So one of the specific aims should be to demonstrate the efficacy of the treatment in patients with disease. It doesn't have to mention the exact endpoint, or it can. You don't have to, you may or may not want to mention the hypothesis, but the point is, many specific aims do not have corresponding trial objectives. The primary trial objectives should have a place or a home in the specific aims seeking support for that trial. Okay. By the way, I looked online for this material. I will tell you, this is not clearly laid out in places. I want to spend a second on review material for the grant application, review criteria. So the review criteria over years have changed, and they've changed not in intent, but in structure. And these are the so-called simplified grant review criteria for NIH. I think that we're in 2024, judging from the URL at the bottom. So the review criteria are primarily divided into 3 factors. The first is the importance of the research. So that is importance because the question is important, the gaps are important, and if you are successful, you will be able to plug those gaps. The second factor is innovation. Are you generating experience with new ways of doing things that will be generalizable and therefore further other investigations beyond yours because you're bringing innovation to the field? So innovation is not innovation just to be tricky. Innovation here is because you're furthering the art of science or investigation. The second factor is rigor and feasibility. Can you run the trial, and when the trial gets a result, do we have a high level of confidence that there's both good internal and external validity? Meaning the answer you got accurately reflects the experience in those patients, that's internal validity. So that means no loss to follow-up, blinding, lack of bias, those sorts of things. and good external validity, meaning the answer that you've achieved also applies to other populations and settings outside of your trial. So that means, for example, you've sampled from a population that is typical or representative of patients suffering from the disease. The third factor is expertise and resources. This is, do you have the necessary training, supervision, institutional support, etc., both among the personnel and in the environment, so that you'll be able to successfully pull this off. The point I want to make here is that the information that's in the clinical trial protocol is key material for evaluating factor 2. So one of those three scored areas is really directly addressed by the protocol. So if you have a clinical trial protocol separate from your grant application that is in good shape, and it can be included as an appendix to your application, then it can greatly improve the ability of the interested, for hopefully the primary reviewer, to verify their positive assessment of factor 2 is in fact justified by the details in your clinical trial protocol. Okay? So bottom line 1, these documents are so different that very little of the text in either document can be repurposed directly for use in the other. So this is an admonition against copy and paste. If you do that, as a reviewer, first, yes, we will notice, because we'll realize what will happen is we'll say, oh my God, did I fall asleep? I seem to be reading the same thing again. And then we'll flip around and we'll realize, yes, we're reading the same text again, and then we'll be irritated. Second, it's going to seem sloppy or lazy, and you will make mistakes. You will either duplicate information that doesn't need to be there, or you will leave out stuff because it wasn't important in one of the documents, but it is in the other. So try not to write these two documents at the same time and sort of copy and paste and modify stuff between them. Bottom line 1.1, so a little extension of bottom line 1, is that there are sections of the grant application that really have no corresponding section in a clinical trial protocol. The detailed scientific background, the specific aims, other than the connection I mentioned earlier, the progress report, your leadership plan, contractual agreements, budget and justification, letters of support. So all of your work developing those sections of your grant can occur in parallel, before or after, but independent of the writing of your clinical trial protocol. Okay? The flip side is that there are sections of a clinical trial protocol whose sections are only very briefly summarized in the grant application: adverse event reporting, some data collection procedures, maybe even some patient enrollment or identification procedures. So they are mentioned in both documents, but in markedly different detail. So a common question, I have to say, this is something that the instructors for this course have debated, is whether you should try to write your grant application first and then develop a clinical trial protocol for it, or the other way. And I will say that there is not consensus, and people do different things. So it really is a matter of personal preference. But like many things, whereas there's personal preference, I do have an opinion, which is that I think writing the clinical protocol first, it puts you in a much better position, especially for someone who's relatively early to getting funding for multi-center or substantial clinical trials. And the reason for that is that when you write your clinical protocol, you got to think about all the nitty-gritty details. Oh my God, I can't ask them, this question, or I can't collect this data point, or I won't have this when they're randomized. How am I going to use it as a stratification variable? You think about all those details, you write this detailed document, and you've sort of thought about everything that you can at that point. It is much easier to take that level of detail and simplify it to a description to go into your grant than to take a broad, sort of hand-wavy description of your clinical trial and expand it to a protocol. I just think it's a lot easier to cut than to add. Second, when you're thinking about all those details in your clinical protocol, you're just thinking about, can I assess this endpoint in a way that I'm still blinded to the intervention, or will I know what they're getting? Will the ICU nurse tell me whether or not they've done something that's related to the trial intervention? Helping you think about all those details ahead of time, or having you think about all those details ahead of time, will help you write about the merits of your approach in the grant application with much greater clarity and precision. You can add little examples of what you mean. We will take we'll take tremendous care in ensuring blinding of the outcome assessors, for example, and then you pull something from your clinical trial that shows you've thought about exactly the details of how this is going to work, as opposed to just asserting that, yeah, you're going to pay attention to it. Those little details greatly improve your credibility to the grant reviewer because you've clearly thought about the details. And then third, by having the trial protocol first, not only can it be included as an appendix in a grant application, which I think is very valuable, but you have it for your IRB submissions, for regulatory purposes. It's sort of the anchor point from which everything else can flow. Now, that said, this is a matter of personal preference, but if you have time to sequence this either way, I think this generally works better. Now, there are some pitfalls to the fact that you have two documents, and I call it poor document hygiene, which is essentially inconsistencies between descriptions of parts of your trial that really should match up. So that can be inconsistencies between the grant application and the clinical trial protocol. I recently was reviewing something where one said the maximum sample size was 146, and one said the maximum sample size was 147. It was literally because two people had rounded something two different ways. And that inconsistency makes you think this person does not have their act together. They're in a hurry. They don't have this precisely defined. So you want to avoid those inconsistencies. You want to make sure there's no inconsistencies in the statistical design and analysis between three documents, all of which will describe it to differing levels of detail. The grant application with the least detail, the clinical trial protocol with medium detail, and the SAP with the greatest level of detail. Now, inevitably, there are little things that are different. And it is useful to define a hierarchy of documents, especially for the statistical analysis, so that in the case of discrepancy, it is unambiguous which one takes precedent. And the usual hierarchy is for the statistical analysis, the SAP is the final say versus the protocol, and the least weighty is the grant application. Okay. I will tell you in review work that I do for journals, I always look at the SAP first. And then when I'm reading the manuscript, I note every place they say something in the manuscript that's different than the SAP. And every one of those is a possibility that they sort of played with what they were doing to get the answer they wanted, decreasing the reproducibility or the validity of their research. Now, once you start your trial, it is very common to find things that you wish you'd done differently. So you want to try very hard to maintain version control and consistency as the documents are updated. So to the extent possible, if you don't copy everything in every place, it reduces the number of documents you have to update when you make a change. Okay. Couple of things very quickly. The statistical analysis plan is this detailed document of the statistical plan. I did find this article that includes a senior author, Jason Connor, a colleague of many of ours, that they actually provide an outline for a statistical analysis plan. It's a pretty detailed outline, but it gives you a place to see all the things you might want to fill in. Things that people sometimes miss is the definition of the analysis populations, the missing data plan, those sorts of things. Similarly, the manual of operations, the manual of procedures, sometimes called the manual of operations or MOO, is this dynamic document. It is not required for all trials, but if your trial has complicated procedures, it may be. There's a nice Word template of it on the National Institute of Dental and Cranial Research site that, again, it has a lot of detail, but it provides you an idea of what these documents look like if you've never seen one. I do want to point out that when you submit a results manuscript for your trial, the peer review process should include an evaluation of your original trial protocol and your modified version. So please keep a time-stamped copy of your trial protocol each time you modify it. Same for the statistical analysis plan. And then depending on what happened in your trial, the journal or peer reviewers through the journal office may request or want to review your DSMB charter or the interim analysis reports. So get in the habit of saving everything in a time-stamped way. So if anybody asks you, what did you know on a certain date, you can answer that question without difficulty. When in peer review, we ask for any of these documents and the person just sends in the grant application, that decreases their credibility. Okay, so in summary, these are all different documents. They have different purposes, different content, different audiences. And so you want to, when you're developing any of these documents, have clearly in mind what the purpose and audience and content should be. And I think as a general rule, don't try to don't try to work an hour on your grant and an hour on your protocol, but try to get one in pretty good shape and then shift your mindset over completely to working on the other one rather than trying to multitask. Thank you very much.
Thank you, Roger.
Yeah,
All right.
Roger, that was great. I just want to second the discussion about how it was hard for us to figure out what we should tell people to work on. And part of the motivation for the protocol is really there's plenty of grant writing courses, but there aren't very many places where people really can help you develop a clinical trial protocol. So, this doesn't replace a grant writing course per se, but you did a phenomenal job of comparing and contrasting the two. So I saw a hand go up, so I'll be quiet now. But.
And I do want to say I didn't use the template that Megan provided to me, and the last slide is supposed to say, please fill out the evaluation.
That's also in the chat, and
Yeah.
and Megan can put it in for those who joined late as well.
Great, thank you.
Questions for Roger?
You can put questions in the chat and I'm happy to read them, or you can unmute and ask.
Hi, Roger. This is Manuel Schindler. This is something that came up when you talked about making sure that things are consistent among the documents. I've been dinged before because sometimes when I talk about the expected number of subjects I'll need, but then somewhere else I talk about how many I have to enroll, because you always want to add 15% or so and so I've been dinged before for citing different numbers. But how do you maybe that was just a reviewer who didn't carefully notice I was talking about what my expected final number was versus how many I have to enroll. Is there what's your recommendation there in terms of clarifying enrollment versus how many subjects you actually need?
Yeah, so I think there's usually 3 numbers. There's the number that you need to screen in order to reach the enrollment. Your enrollment is the number that's defined by your sample size calculation. And then because there is going to be, as a general rule, some patients lost to follow-up or who drop out, there's a number of completers or for whom the final assessment is. And you want very early to use some consistent terminology. So we often use, screened, enrolled, completers, screened, enrolled, final sample size. And you just want to use the same terminology in all your documents and generally put them together. If you have room, depending on the format of the grant, to have a little sort of mock consort diagram, so you show the dropouts, that can also be helpful. And then it's possible that this is also an example of reviewer fatigue. So depending on the setting, reviewers often have lots of grants. None of us ever allocate the time that we should for every task that's assigned to us. And so people are often reading quickly, and reviewers will sometimes make mistakes where they just didn't catch a distinction because it's in one place but not everywhere. So you do want to write these documents to make it as easy as possible for the reviewer to get it right the first time. And as a general rule, if you spend a few more words sort of simplifying something, that actually works more in your favor than spending those few additional words to add an additional technical detail.
Okay, thank you.
Yep. Other questions?
I have a question, actually.
Okay.
That's okay. When you talked about establishing the hierarchy of documents, I thought that was very interesting. And where would be the best place to define that hierarchy?
Yeah, so you want the hierarchy to be explicitly listed in the places where it's likely to be applicable, and by far the most likely is in the statistical analysis section. So I kind of like the last sentence in the statistical analysis section of the protocol to say that there will be a separate statistical analysis plan document, and in the case of any inconsistencies, the SAP takes precedence. And then in the SAP, you write in the introduction section, this document takes precedence over other documents in establishing the pre-specified statistical design.
Thank you.
Yeah. And it's just a hard learn, this is a hard learned lesson when people have been asked, why are these two things different? And they didn't realize they were different. They swear about the reviewer, and then they don't know what to do. And if they just put this in, then they just say, oh yes, we have a plan for dealing with our own inadequacies, basically.
There's a question in the chat about who should write each document.
Okay. So I think the protocol and the grant are usually, written primarily by whoever the PI is. And, we expect those to be the students in this course, but in other settings, it's the PI. I think the manual of operations is often best written by a study coordinator who's used to doing studies that are similar, who's really run into all of the nitty-gritty little stuff that can happen. The SAP should be written by the statistician who's teamed with you to design your trial. And the DSMB charter, usually you take one from somebody else who has one and you modify it. Usually, again, it's the PI that does that. But an equally important question is who you should make sure reviews it. So for the grant, you want the grant long before you have to submit it to be read by the types of people who review grants. who will find little inconsistencies regarding what you said you're going to do in the specific aim that is being addressed by a particular part of the research plan. You want grant reviewers to read your grant. For the protocol, obviously it's good to have people who have a lot of experience with the funding of clinical trials, but really you also want your protocol read by people like study coordinators, other faculty who will actually have to do it and find out what their questions are. So you think about the audience for each one of these documents, and the people who review what you write need to be drawn from the audience for which they are intended.
Good question.
Yeah.
Any other questions for Roger? All right, well,
Okay.
thank you so much for joining us. It was an excellent presentation and glad you all could make it. Our next webinar will take place on April 25th, on Friday, April 25th at the same time from 12 to 1 Eastern Time. And the topic will be avoiding pitfalls in early phase clinical trials, presented by Dr. Will Moyer and Chris Coffey.
And the question about will the slides be available, Meg, you're going to post these so everybody has access to them, right?
Yes, they'll go to our website, which is NINDS-CTMC.org.
Great.
Thank
Thank you,
you.
everybody.
I hope you all have a great weekend.
Yep. Enjoy your small group.